Excellent. Sarah, thanks so much for that generous introduction. And I'm so happy to be here with so many scientists I have admired and has influenced my work over long periods of time. Today I'm going to be talking about a recent project in the group, which is probably our most ambitious long-running project to date. And um, I hope I can convince you why it was hard. Because when we set out, we didn't think it was going to be this hard. And hopefully, I can convince you that it's worthwhile and then to go use the data for your own science. The project is called the Reproducible Brain Charts Project. And it is really grounded in kind of why I got into this in the first place. So I'm a psychiatrist. I'm actually in clinic right now, so you're saving me from an hour of clinical work. Thank you, guys. And the overall context here is that psychiatric syndromes are incredibly common. 1 out of 2 people will have a diagnosable psychiatric condition at some point in their life. And over time, the field has really pivoted to a developmental perspective, driven by the fact that mental illness often begins very early in life. And for many like really important and devastating disorders, including psychosis, bipolar disorder, often recurrent depression, anxiety, attention problems, it's now understood as a neurodevelopmental process. So the overall goal of the program of research in my lab is to describe mental illness in terms of variation of patterns of brain development. However, charting normal brain development and variation associated with mental illness requires just massive data resources. And as a result, this has led us over time to focus more and more on methods and data that allows for scale for us to test the hypotheses that we're interested in. And I'll give you 4 reasons to start why big data is required. There's far more than that. But the first is that if we're interested in brain development is a dynamic, nonlinear process. If brain development were a perfectly linear process between, like, let's say the age range we study oftentimes is like mid-childhood, like 7 or so to young adult at age 22. If that were linear development between age 7 and 22, you could just get a couple kids at each year and draw a line through it and you'd understand development. However, we know the brain is a very high dimensional place and development is nonlinear. So we need to sample the brain at each age range with enough density to understand these nonlinearities. So we need a lot of data to draw normal growth curves of the brain. Awesome work from Richard Bethlehem, Aaron, Alexander Block, and many of you guys, like kind of showing the like the best way to do that with structural brain imaging. Second reason we need big data: psychiatric illnesses are incredibly heterogeneous. In medical school, they teach you there's 50 reasons you can come to the emergency department with chest pain. And we understand the chest quite well, in fact. You can have heart attack, sure, but you can also have a pulmonary embolus, or you can have indigestion, or you can have musculoskeletal pain. We understand chest pain pretty well. We do not understand the brain. The brain is multiple orders of magnitude more complicated than the chest, and yet we call depression one thing. There's only one way to be sad, and there's 50 reasons for chest pain. Doesn't make any sense. And so if we want to have any hope of approaching the most important challenges of the field, which is unpacking that heterogeneity that allows us to get to better biology, to develop new treatments and understand prognosis, we just need to unpack that heterogeneity, and it's likely massive, so we're going to need huge data. A third reason that big data is required is that we're kind of caught between type 1 and type 2 error. Brain imaging data is high dimensional. The brain's a big place. So if we just run a ton of tests with standard mass univariate approaches, we're going to get a ton of type 1 error, false positives. And sure, multivariate approaches are one way around that, but nonetheless, you're going to run a lot of tests. We're going to need to correct for a lot of them if we don't want to be vulnerable to type 1 error. On the other hand, though, it's increasingly clear, highlighting this work from Scott Merrick, Brenda Turbo-Clemens, it was out in Nature recently, that the effect sizes we're interested in are relatively small. So a lot of our work focuses on development, and that actually is a big effect size. There's a big difference between an 8-year-old and a 20-year-old. But on the other hand, things we care about clinically, like cognitive variation, kind of small to moderate effect size. But when you get to psychopathology, something you really care about, tiny effect sizes. And there's a lot of reasons for that, heterogeneity, bad instruments, and other things like that. But if we want to kind of proceed with the data we have, we need really large samples to understand these phenomena that have small effect sizes. And the last reason why I'll make an argument for big data is that, in the end, what we want is something generalizable. If I want something I measure about the brain to be useful, it can't be just true here at the University of Pennsylvania. It has to be true everywhere, or at least we have to understand the limits of its generalizability. So we need to have diverse samples that are cover the population at large and help us to make sure they're applicable to all of our patients and the people we might care about. All right, so hopefully I've made the case for why we need big data. But RBC, Reproducible Brain Charts initiative, R is for reproducible, and that's the first letter. So let me unpack that a little bit, and we'll try to make the case for why reproducibility is a must. I think to zoom out a bit, so my lab does neuroimaging work primarily, but I have a lot of friends and colleagues who do other sorts of neuroscience. And if you ask them what their honest opinion of neuroimaging work is, especially people who've been in the field for a while, maybe this has changed a bit over time, but still, and especially if you buy them a couple beers first, they will tell you that most of the time they just don't believe it. They don't believe the work because there's so much, the data is so high dimensional, and then we do so many things to it. So if you have that much analytic flexibility, some scientists might say, is that you just have too much freedom and you can get any result. And this is a real challenge for the field. I think it's gotten a lot more attention over time. But this lack of reproducibility and analytic flexibility is a huge break on creating useful knowledge that actually is generalizable and allows us to make progress. Early in the RBC project, we had a paper together with Mike Millam, who I lead RBC with, just looking at the most basic image processing steps, and not even the later post-processing, just the early pre-processing steps across several of the most commonly used pipelines. And in general, agreement between these things wasn't as good as we would hope. And that is just the earliest stage of the analytic flexibility chain. So this is a huge issue. But luckily, open science provides some pretty clear options on how we can approach this. First is open and track code. And this is, fairly standard at this point now. At least journals will make an effort or kind of a pretense of making you release your code when you submit a paper. But unfortunately, you could have really well documented, well commented, beautiful human readable code, and it's not actually reproducible unless you have the underlying data, though. So really, to have things be reproducible, we need open code, but we need it to be paired with open data. And I really believe that ultimately for a field to make progress, and I'm not the only one who believes this, I'm a relative latecomer to this field. People have been banging on this bell for a long time, is that sunlight is the best disinfectant, and that is the best way to convince our colleagues that our results are real and that they should believe us. So in response to this need for big data and reproducibility, that's where RBC comes in. I lead RBC, as I said, with Mike Millam, who's a dear friend and longtime collaborator at Child Mind Institute. Mike was one of the original progenitors of the open science movement in neuroimaging, starting with things like Indy back in the late 90s. And the overall goal of RBC is to combine multiple large-scale studies of brain development, harmonize the clinical measures, harmonize the functional and structural measures, process all the data, then openly share everything. The raw data, the process data, all the code, all the software, everything, fully open. In the end, the goal is to allow other people to go faster. I have no doubt that the best discoveries made with RBC data will not be from our lab or from Mike's lab or any of the people on the team. It will be by someone else using the data. And that is the goal, is that we do the annoying, low-level, unsexy stuff and make it open and make it easier for everyone else to run faster. I want to emphasize, especially today, when given the news cycle, that this study was funded by the National Institute of Mental Health, and it would not have been possible without generous federal support. So thank you, NIH. So RBC, it aggregates 5 big studies of brain development. The Brazil high-risk cohort, the ColorNest project from China, Mike's two big studies, Healthy Brain Network and the NK Rockland sample. And then a study I've been involved with for a long time now, the Philadelphia neurodevelopmental cohort. In total, this is over 7,000 imaging sessions, 6,000 participants, spanning 3 continents. So I want to be really clear, though, that though me and Mike have been like working on this from the initial conceptualization almost 10 years ago, this has taken generations of students and postdocs and data analysts to push this forward. I want to give a particular shout out to Golia Shafi'i, the first author of the paper that just was posted last week, but also she's the one who put it over the finish line and working really intensely on it for the past year. But there's many people who've been working on this for 6 years together with me and Mike, Natalia Esper, Mauricio Hoffman, Alex Franco, Greg Kerr, Max Cieslak, Taylor Sala. So it's taken a massive team. If you look on the website, there's like 25 people. So thank you guys. It was a real team science effort. I only have a 2-part outline today. The first is I want to talk about some of the challenges and opportunities that we've had to face with RBC, and the second is some initial applications. The challenge is, and this is what I said at the beginning, what I was trying to convince you is RBC was way harder than we thought it was going to be, and I want to talk about 5 things we faced that hopefully you won't have to face if you want to use the data, and also have allowed us to learn and make progress going forward, and hopefully make it easier next time when we want to kind of pull in a lot of new data. This spans for everything from psychiatric phenotyping to data access and a lot of nitty-gritty image processing in the middle too. If you tune out, it's okay. Can come back for the next challenge if one of these challenges is not your cup of tea. And also definitely put questions in the chat. I'm happy to talk about this at any time at length. So challenge number 1, variation in psychiatric phenotyping. Psychiatry is a long ways behind the rest of medicine. If I were to see you in clinic this morning with life-threatening mental disorder, I wouldn't be able to send you for a scan or some blood test or something like that would guide prognosis or influence treatment usually. Most of the time, it's just me talking to you, or usually there's one of the residents. And sure, we might spend more time with you as a patient, but in any other field of medicine, if you didn't order diagnostics, you would run screaming for the medical board. And the challenge with this sort of diagnostic procedure is that we're reliant on diagnoses that are defined by the DSM, Diagnostic and Statistical Manual, which defines what we can diagnose people with. And it is well known and extremely well described that the DSM does not carve nature at the joints. So if you have, it doesn't reflect real biological categories. So if you have one mental disorder, you are likely to have more than one. Comorbidity is the rule, not the exception. And furthermore, beyond this like massive comorbidity and non-specificity of our diagnoses, like the DSM ignores any dimensional effects of disease severity. If you have like a little bit of depression, that's very different from life-threatening like depression that could end in suicide. In response to this problem with the DSM, there's been a lot of efforts to try to get to a better framework that reflects the clinical reality in front of us. The High Top Consortia is one of them, which is proposes a dimensional hierarchical structure of nested levels of symptoms. I can come back to this later. But even if you adopt a dimensional structure like High Top, it's challenging when you're doing something like RBC, because let's say you have a measure that's collected in different places, just like scanners are different, psychometricians will tell you that different instruments behave differently in different settings. And the response propensity of someone filling out the CBCL, one of our most common instruments, might be different in Brazil than it is here. So we need to like kind of face some like basic psychometric facts if we want to approach something like RBC. But even more challenging is, what if you don't use the same instrument? How do we combine clinical data if we have studies where things are assessed by different instruments? And I would say This was the part of RBC that intimidated me the most because I'm not a psychometrician. But luckily, we had a crack team of psychometricians, including Mauricio Hoffman, Giovanni Saloom, and Tyler Moore, who's been with us from the beginning. And in a series of three, but now more papers, Mauricio, Giovanni, and Tyler solved these problems, or at least provide an initial solution for these problems. So the first potential problem is the obvious one, is that different studies use different measures. So the PNC, the Philadelphia Neurodevelopmental Cohort, collected a pen. We use kind of a unique instrument. It's a very good instrument, but no one else uses it, called GoAssess. Every other study used the Child Behavioral Checklist, the CBCL. How do you combine that data? And then for all the different studies that use the CBCL, how do you make sure that the psychometric properties of things like invariance are the same across the different samples? Using a combination of item response theory and different factor models and a lot of psychometric magic, they came up with a harmonized bifactor model. And a bifactor model, let me briefly explain, it kind of is in the mode of the high top consortia because it provides a hierarchical structure of nested symptoms. The first of which is something called the P factor, which basically represents overall psychopathology. So if you think back to Psychology 101, you probably may have learned about, if you took that class, you might have learned about something called G intelligence, or Gardner's intelligence. And that responds to the idea that, like, in general, if you're smart at one thing, you tend to be smart at other things. And that's true in psychiatry too. If you're sick in one way, you tend to be sick in many ways. There's an overall liability for psychiatric symptoms. And so a bifactor model, what it does is it extracts this overall factor and then also fits orthogonal subfactors. And this is really nice because then you have this overall factor and these subfactors that are orthogonal, so you can model them jointly very easily in the data, and it kind of gets around this really tough comorbidity problem. As part of this work, Mauricio and co tried like 11 different bifactor models and tried to make sure which one was like fit the data the best and was the most invariant and generalized the best across sites. And in the end, we landed on a factor model first proposed by McElroy, which included an overall factor and then an internalizing factor. That's things like mood and anxiety symptoms, externalizing, which is like conduct and disruptive symptoms, and then an attention factor. So an overall factor and three subfactors. And if you want to see this, like why this matters, you just have to look at the comparison between the PNC, this study collected at Penn. It's a community sample, so kind of a convenient sample. It definitely has some sick kids, but there's a lot of healthy kids in there too. And then the CBCL data from HBN. So different instruments, different places. And the key thing is, unlike the PNC, HBN is a self-referred clinically enriched sample, so much more sick kids. And if you look up here at the top panel, that's the original model score. And you'd say, okay, well, it's reasonable, similar distribution between the studies. Like you would conclude that the PNC and HBN had around the same level of severity of psychiatric symptoms. Because again, this is the overall psychopathology score. After harmonization with item response theory, you can see what the reality is, which is that HBN here in yellow has much higher levels of psychopathology, and whereas PNC has much less psychopathology over here. So this is a way of seeing that the harmonization worked between the different samples. Another way is just like look at how these factor scores fit to different diagnoses. So for example, you have the P factor here in green, and then you have the subfactors here, for example, like internalizing in brown. And you see kids with anxiety and depression have high levels of overall psychopathology in green and high levels of internalizing symptoms in brown. And in contrast, kids with conduct disorder, really disruptive behavior disorder, they have a lot of externalizing symptoms here in pink, as well as overall symptoms in green. In contrast, healthy kids, low symptoms across the board. So this is a nice dimensional framework that pulls in all these different instruments and different populations and allows you to analyze them together. All right. So that was challenge number one. Challenge number two. I'm banging on about reproducibility here. How do we actually make this fully open and convince everyone that what we did is valid and show them all of our work? And this is complicated because our image analysis workflows are pretty complicated. This is just one section of a image analysis workflow we use every day, but there's many sections like this linked together across software packages. And if you were to say, how do we normally keep track of these sort of things computationally, you would say using Git. Like that's how almost everyone does version control of their code, whether through GitHub or Bitbucket or something else. But Git is the industry standard for version control of code, and you can see who changed what lines, who added what over time. It allows all sorts of branching and merging. It's awesome. How do we do that for data? The nice thing is, just like Git allows you to version control your code, something called a Git Annex allows you to version control your data. And Git Annex was originally developed in the context of file sharing for music, funnily enough. And it basically allows you to track every change to your data on a bit-by-bit basis, just like Git allows you to track every line change to your code. Git Annex is challenging to use, and over time, software called DataLad has emerged to allow us to work with data in Git Annex and have an audit trail of any changes to our data, just like Git allows us to track changes to code. As we were starting RBC, work from Adina Wagner, Michael Henke's group, introduced what is, I think, still the gold standard for reproducible image analysis workflows with data labs, a workflow they called Fairly Big. And all of the image processing that we did with RBC used Fairly Big, but I will say it was a pretty steep curve that we would have not gotten up this curve without the real collaboration and help from Adina and Michael and their whole team. Data Lab folks are just awesome. But we found it hard enough, and our informatics team has a pretty high technical skill level, that we wanted to make it easier for everyone else too. So Qinying Zhou, a awesome bioengineering PhD student who's now a senior engineer at GE China, developed something called BABS, or the Bids at Bootstrap. And this allows for an automated, fully reproducible implementation of the Fairly Big workflow without all of the technical headaches that come with like climbing the learning curve of DataLad, which is quite steep. And so even though we didn't use BABS for RBC, we used kind of standard Fairly Big. Going forward, we use BABS for everything. And what BABS allows you to do, all it needs is a BIDS data set that's like the standard image, data structure for imaging, BIDS, and then a container that does image processing. So just as long as you have those two things, you can use BABS, and we use BABS all the time with many of the tools that we develop in the lab. Things like fMRI prep for functional image preprocessing, and then XCPD, which takes fMRI prep output and does post-processing on it. So it was developed originally by Roscoe Chirich, and it was a really very significant update by Kahini Mehta and Taylor Salo last year. For diffusion imaging, I'm not going to be talking. We do a lot of diffusion imaging in the lab. I'm not going to talk about it today, unfortunately. But QSI Prep is software that's really widely used, developed by Matt Cieslak, and just awesome, really actively maintained and developed. And then ASL Prep for arterial spin-labeled MRI to look at brain perfusion, developed by Aziza Mbimbe and now maintained by Taylor Salo. Also, if you ever want to try these tools and things don't work, we always want your feedback. Definitely let us know. We want to hear not just the success stories. All right, challenge number 3. How do we curate this massive collection of heterogeneous imaging data? Because imaging data, like different studies, acquire different image types on different scanners at different places, and sometimes even changes over time in the middle of a long-running study. So it varies across studies, but even imaging data varies within studies. There's a lot of, even though in a paper you might say, we acquired, this sort of data, and you list out the parameters, usually that's like might describe like 80 or 90% of the data. But then 10 or 20% of the data, something weird happened, and most of the time we're not even aware of it. However, the rise of BIDS, that's the brain imaging directory structure, has made it more easy to kind of get in the weeds on this. And BIDS is really the field standard data structure now, and allows for standardized data organization, standardized naming conventions, and really detailed metadata. However, one challenge is that for many studies, including PNC and HCP studies and ABCD, BIDS wasn't adapted by the consortia at the beginning and isn't released often as BIDS. Another awesome thing about BIDS is that it allows for BIDS apps. And a BIDS app basically reads the metadata in BIDS, and then the software auto configures to apply appropriate preprocessing. So you don't have to say, oh, I'm going to tailor my pipeline for this edge case where we didn't get a field map or something like that. And BIDS apps are awesome. All the software I talked about in the previous slide are BIDS apps, but it's a real kind of gotcha that people forget, which is that BIDS apps depend on correct metadata. And if the metadata is incorrect, the image processing can be what we call reproducible but wrong. Like it reads the metadata, but the metadata is incomplete or wrong. The pipeline configures having read that metadata, and then does something silly. So this really kind of points to a hole in some of the curation tasks that we have. And when we get 6,000 participants worth of images, and each participant has many different image types, the first task is like, what data do we even have here? Like, what's in the box? Second, like a very common curation task is, well, these are just too weird. We're going to exclude those. We're not going to include them in our study. We want to make sure everything is in BIDS format and valid according to BIDS validator. We want to first identify and then fix missing or incorrect metadata. And then we want to usually, the best way to do it is, okay, if we have this amount of variation, we want to test our preprocessing on a subset of scans that covers the full amount of variation. And when we started RBC, there was no tools to do this, to do reproducible image curation at scale. And the reason this matters is that most of the time, these curation tasks, they're like done by one usually underappreciated graduate student or data analyst or postdoc who keeps some like notes in a Word file or something, and then we don't really know how these decisions were made. And this is a problem because if it like breaks the chain of reproducibility right at the beginning. And like if you don't have reproducible image curation, how you get from this online data resource to your analytic result, you're like lost right when you leave the door. So to address this challenge, Sydney Kovitz, Matt Cieslak, and an awesome team developed QBIDS. And QBIDS is a awesome tool for image curation. It allows you to see what's in your data. It allows you to make decisions about what to include, what to exclude, how to rename things. It integrates the BIDS validator. And critically, it's all fully reproducible because all the steps integrate DataLad. And I think probably the strongest advertisement I can give for QBIDS is that I've been working with the PNC data since I was a postdoc in 2008. And until we had QBIDS, I didn't really understand how much heterogeneity and kind of weird stuff there was in PNC. And PNC is an awesome, clean study compared to a lot of things that are out there. So if you want to get to know what data you actually have and you want to curate it reproducibly, QBIDS is great. It was a complete workhorse in RBC, and we wouldn't have been able to do it without it. All right. Challenge number 4. Sarah referred to this, and this is I find this both gratifying and a little bit cringy, is that when people meet me, they usually think I'm like Dr. Motion Artifact, and I guess that's true. And I think the reason it stays true is because motion just remains this like incredibly pernicious bias in all imaging data. Going back to when I was a postdoc, 2012, synchronously with Kuna Van Dyke at Randy Buckner's group and Jonathan Power, Steve Peterson's group at WashU, we all described that motion artifact has this incredibly strong influence on functional connectivity estimates. This is particularly important for studies of brain development, though, because I've got 3 kids, but you don't have to spend much time around kids to recognize that kids just don't sit still. And the problem with motion artifact is the effect is so strong in the data that if you want to study brain development and you do not account for motion artifact, you will not be studying brain development. You will just be describing technical variation in the data because kids move. And that's not just true for development, that's true for psychiatry too. People with higher levels of symptoms move more. It's true for all sorts of different medical disorders. Data quality remains the strongest influence we have that we can identify in our data. Technical variance outstrips biological variance every day, so we have to be really, really careful with it, especially when it's correlated with biological variables that we are interested in. So to get around this, a huge focus in RBC was on data quality and harmonization of data quality. Because this gets back to kind of who you include in your sample. Like if different people, like let's say you have a data resource and different groups apply different sorts of quality assurance, that's for inclusion criteria. So you're going to start out with a different sample, and then you might adjust for quality differently across these different studies. You're definitely going to get a different result because you're like looking at a different subset of the data and then controlling for data quality in a different way. And this is hard often, like especially when you're trying to aggregate different data, because unless you're willing to start from zero and do it all fresh, usually it won't be harmonized because different groups do data quality curation in different ways. So we bit the bullet with RBC and did a really extensive amount of data quality curation for both functional and structural data. And you can see here in panel B, the different studies have a very different level of kind of data that we think is unusable. So, for example, HBN, which is the sickest kids, unsurprisingly have the highest proportion of fMRI data that's probably unusable. And just like we've seen previously, and a lot of people described, that motion declines across all these different data sets as kids grow up and younger kids move more. So I've been talking about functional connectivity, but we know that in-scanner motion also impacts measures of brain structure. This is work from Martin Reuter and Dylan Tisdall showing huge effects of motion on cortical thickness. Unlike functional data where you have an objective measure of how much kids move when you're acquiring your fMRI data and how much motion there's in the time series, structural imaging data is less clear for most acquisitions. Many scans that now have navigators in them, you get an estimate of motion, but a lot of the data sets that we included did not have that technology when the studies were run. As Sarah mentioned at the beginning, we do have an automated measure of data quality called the Euler number, but the Euler number is it's a standard output from FreeSurfer. It measures the topological complexity of the cortical surface, and in general, you get more holes in your cortical surface the noisier your data is. The problem with that is there is significant variation across acquisition scanners, and the Euler number itself is hard to harmonize. So in order to harmonize the Euler number, we needed to come back to some ground truth, which was manual expert rating. So all of the nearly 7,000 images in RBC were rated by 3 expert raters. It was a really intense quality control procedure where we first had this lead-in period where all the different raters had a training data set and they had to establish reliability that were supervised by expert raters. And then we set them loose and continued to assess reliability among the raters as we went. And what we found is just like for functional data, the data set that has the highest proportion of kids with significant clinical symptoms, the healthy brain network data, had the most images that were affected by data quality. Just like functional data, in general, as kids grow up, their image quality improves. All right. Enough on data quality. The 5th challenge here. This, to anyone who's ever had to use a open access data set, and then questioned how open it really was after spending months trying to get access, this one is for you because it seems obvious, but open data is only useful if you can access it. Many studies in the US were required to deposit our data at NDA or the National Data Archive. And NDA is in many ways great. It is not perfect. And one way it is not perfect is the requirement for a data use agreement. I'm not sure how many of you guys have ever tried to get data from NDA, but it requires a university official. Then it's got to be renewed annually. Adding any new rotating student, you have to do a new data use agreement. And then there's all these different layers of approval and review. So getting access to quote unquote open data often takes months. This is a significant frustration to many scientists, and it's just a real brake on progress. If you just think about each of those delays for each of the groups. So one of the goals with RBC was just to make it as open as possible. And uh, like one of the things is that as we were selecting what data goes into RBC, we had a lot of negotiations with sites, and we had to choose which data sets to use to allow it to be really open. So RBC is privacy preserving. All the structural images are refaced. So that's like kind of not just defaced, but we actually register a template face to the brain. So you can't see whose face it was, which would in theory allow you to identify someone. It only includes summary measures of psychopathology, not like granular measures that are potentially identifiable, and IRBs would not let us share. But because of this, because we will use anonymized images and summary measures of psychopathology, it allows us to share it truly openly. So it's on INDI, the International Neuroimaging Data Sharing Initiative that's started by Mike Millum, close to 20 years ago. And through his partnership with Amazon Web Services, or AWS, Amazon allows us to place hundreds and hundreds of terabytes of open data on AWS for free without upload or download fees, which can really get you. But the caveat is it has to be truly open. And so that's the nice thing about RBC. It is truly open. You don't have to ask me to use it. You don't have to email me. You don't have to talk to Mike. You don't have to fill out a data use agreement. You don't have to get a university official to sign it. You just go to AWS and download the data, and you go. And because of that, there's been really rapid uptake of RBC. This was from in like with the download curve in April when we like sent our first tweet about it ahead of OHBM, and it's just continued past then. It's I think over 1,000 downloads already. So if you want to use RBC data, I would go to the website. It's you can scan it here, but it's reprobraincharts.org. We have a lot of information on how to access the data, how like links to all the technical papers describing the tools I've mentioned. And if you run into trouble, you can post stuff on Neurostars. We have a really active presence on Neurostars, especially Stephen Meisler, an all-star postdoc in my field. I think he's like responds to 80% of the questions on Neurostars. And just let us know what your experience is. We've spent a long time on this. We want people to use it. Let me just check on time before I move to some of the applications. All right, we got time. Excellent. So an initial application that Golda Shafi'i, superb postdoctoral fellow in my group, who wrote the paper associated with RBC data release and is now in bioRxiv, we wanted to show kind of why this stuff matters. And as some of the toy examples that we pursued initially as part of this data release paper was to say, okay, well, what happens when you don't harmonize the data and when you don't adjust for things like data quality? So here you can see the 5 studies. Here you can see the data, just like approach naively. And you can see that, yeah, you can see that on the x-axis you have age 6 to 22. On the y-axis you have the mean cortical thickness. And you can see that, okay, yeah, there's a clear trend here. Like we know cortical thickness declines with age, and indeed you can see this in just like the raw data off the like, kind of naively approach data off the shelf. But if you look at the trend lines for each of the studies, they don't line up all that great. However, once you apply this really careful quality assurance and harmonize the data using tools developed by our awesome collaborator, Taki Shinohara. In this case, we used CoVBAT, which is kind of the next generation of combat, using that also allows you to use a generalized additive model to covary for nonlinear effects. You can see that things line up just much, much cleaner. So this is hopefully one reason why you should use RBC. Also, it's just way easier. A similar thing that is apparent for functional data. If you approach the data naively, you actually get more results than if you do careful QC and harmonization. That's not surprising because we know that data quality is worse in younger kids. So if you don't account for data quality, you're going to find all sorts of things that you think are development, but are in fact motion. And so kind of two results I want to highlight is that even in the subset of results that is survives after careful QC and harmonization, can easily reproduce some of the most common findings in the literature, including increased connectivity within the ventral attention network, increased segregation or decreased connectivity between the default mode and ventral attention network. And again, I just want to thank the huge team that made RBC possible. But in the maybe 8 minutes that remains before I pivot to questions, I want to talk about one application that data like RBC allows you to do, which is some of the major focus of work in the lab, which is hierarchical cortical development. And this responds to the fact that cortical hierarchies are really critical for understanding cortical development. And to understand the hierarchies, you also need to understand development. They really go together. This goes back to canonical work by Marcel Mesulam, who described that the brain is organized into large-scale neurocognitive networks to support hierarchical information processing, with primary cortex like here in blue, with highly defined layer structure and canonical feed-forward circuit structure, and transmodal association cortex, like for example in pink, less defined layers, less canonical circuits. However, since 1998, the field's come a long way, and several years ago, just phenomenal scientist who I think talked at this series, Valerie Sidner, when she was with a graduate student with me before. She's now a postdoc at Pitt with Bea Luna. I wanted to look at brain organization across feature types to see if there's a common hierarchy of brain organization. And indeed, she found that across all of these different types of features, whether it's the evolutionary expansion, cerebral blood flow, gene expression, activation to tasks, cortical thickness, different metabolic measures, she saw the same hierarchy again and again. And then she integrated this to create something that we now call the sensory motor to association axis. This is kind of a way of looking at the brain and kind of the common organizational structure that's been incredibly useful for understanding development and also many other aspects of brain organization in health and disease. And the sensory motor association axis spans from the sensory motor pole here in yellow. I can see in like visual cortex and somatomotor cortex. These are sensory motor part, and then the association pole is an association cortex here in purple. And this links to things that different functions of the brain, like sensory motor cortex is responsible for things like action, perception, and sensation, whereas association cortex is important for things we care about in psychiatry often, like executive function, mentalizing, and social cognition. And one major take home, if you were to say what have I learned over the past 10 years from studying brain development, I would say that the main take home is that development both aligns with and refines this sensory motor to association axis of brain organization. This was quite clear in work from an awesome grad student at the time, now a postdoc at Stanford, Adam Pines, who showed that that connectivity between networks diverges with age. So if you assign each location of the brain to a network and then just ask a very simple question, how connected is it to networks other than its own? You can see that here in yellow is the association cortex, here in purple is sensory motor cortex. In general, sensory motor locations have more coupling to other brain networks early on, whereas association networks have less. And then this diverges with age such that you get this greater span as kids grow up. And so another way to look at this data is if you look at where a network is in this functional hierarchy defined by the sensory motor association axis here on the x-axis. So this is association, the association pole here. This is the sensory motor pole over here on the x-axis. And on the y-axis, we simply have the age effect. Like how do these networks change as kids grow up? You see this incredibly strong relationship such that where a network is in this overall pattern of brain organization largely explains how development occurs. This was such a strong finding that we wanted to use data like RBC to really check how generalizable it was. And this is work led by Audrey Lua, a fantastic senior MD PhD student in the lab, who took this initial result in the PNC, what Adam found, and then replicated it in 3 independent data sets after pre-registering everything. She took the step of writing it all down and then just did the thing in 3 more data sets. And what she found is incredibly convergent results showing that the development of functional connectivity really aligns with the sensory motor association axis. So, for example, you can see in all these data sets, sensory motor networks in general, they have, again, on the y-axis we have functional connectivity strength, so that's overall connectivity. You can see that it increases in sensory motor networks, kind of in the middle of the axis, and then it declines at the association pole networks like the default mode that sit at the far end of the association pole. Another way of looking at that, same plot as I showed you before. So on the x-axis is the location of a given region on the hierarchy. So over here, this is the association pole of the hierarchy. This is the sensory motor pole of the hierarchy. And you can see that in all of these data sets, you have this very predictable relationship such that in general, we get more connectivity with age as kids grow up at the sensory motor pole, and you get less connectivity at the association pole. You get more network segregation. And this is true across all these different data sets that were acquired at different scanners, at different locations, at different times. And if you look at the paper, there's like a million supplementary figures with under different pre-processing conditions and different parcels and everything else like that. So we just cannot get rid of this result. It's incredibly strong. If you want to unpack that a little bit, because that measure on the y-axis was overall connectivity strength, what you can see is that if you look at, again, on the x-axis, we have where a region is in this hierarchy, and same on the y-axis for like pairwise connectivity. And then in this topo map, yellow is increases in connectivity with age, and purple is decreases in connectivity with age. You see this incredibly reproducible effect across all 4 data sets. And so anytime, to come back to where we started, anytime I am faced with someone who's skeptical of neuroimaging data and wonder how believable it is, I point people to this paper, which just shows incredibly strong effects that Audrey wrote down the analysis ahead of time. So just to recap, maturation, functional connectivity aligns with the SA axis. Development serves to refine, enhance feature variation across this axis. And this is really driven by sensory motor networks integrating and segregation of association networks. You know, kind of reverse inference on imaging data is always challenging, but if I were to speculate, we could say this developmental pattern could allow for both ongoing motor development and specialization of higher order cognitive systems. A lot of the work going forward that I unfortunately don't have time to get into is to try to understand what are aspects of developmental plasticity that govern the development of cortical hierarchy. There's like a whole series of papers led by people like Val Sidner and Bart Larson that get into this. If you want a teaser, I would check out this review they had in TINS on a critical period plasticity framework of SA axis cortical development. And with that, I think I'll leave a couple minutes for questions. I want to end by thanking people who actually do work in my lab. You know, I'm front of the house in sales and giving talks to wonderful people in the UK. But you know, the students and the informatics team are the people who just day after day just do amazing work. I want to especially Matt Cieslak, Audrey Lua, Gola Shafi, Taylor Salo. A lot of the alumni are moved on to bigger and better things. All of our collaborators at Penn and Chop. Mike Millam, kind of like really strong mentor and has had a lot of influence on my career and like really was a huge intellectual force behind RBC. And again, I want to end by thanking the federal government for their generous funding over the past 5 years.